Well, thank you very much for accepting our talk in this very interesting session. So today I would like to share some early results of some colleagues and I in an experiment where we try to use the R package biosurvey to guide paleontological surveys towards unsampled climates. So first of all, why are we interested in paleo niches? Because the objective of our group is to work on paleo niches. So just let me, okay, that's better. So project, it's very useful to use to think about paleo niches because we can project modern niches to the past that may reveal past distribution and dispersal trajectories in response to past climate change. So for example, if we take the ecological niche modeled from chimpanzees in the present day, we can find like some different distributions that may have been suitable for the chimpanzees in the past. But, okay. But the problem is that the vast majority of paleo-ENMs are not used to model fossil species niches distribution, but rather to project modern niches, as shows this very interesting graph from a recent paper that was surveying what kinds of occurrences were used to model paleo niches. And as you can see, most of them, thinking in the past, don't use any fossil data. But we actually need fossil occurrences to be able to model fossil species niches. Why? Well, because um, the niches are changing at the million to tens of millions year scales, and many fossil species have no modern equivalents. So that's why using fossil occurrences may inform us on past fossil niches better than actually projecting back modern niches onto the past. So it can allow us to think about effects of orbital control and climate on potential ranges. We can also test some dispersal hypotheses, etc., etc. So now the aim of our paper was basically to think about how can we have a better occurrence data of fossil species. So first, let me run to this how the correlational niche modeling works. So to model a species niches, basically we use the geographic distribution of a species, and then we run a model, and we can basically reconstruct the species ecological properties in environmental space. Then afterwards, we can project that onto the geographic space and then get those very nice maps where we have the potential distributions or the suitable habitats of the species. The problem of distributional data is that the abiotic variables, so the fundamental niche ecological properties of the species, are not the only variables that impact the distribution of the species. We also have biotic variables, so meaning the interaction with other species that is going to constrain or to expand these distributions. And we also have related to the dispersal capacity of the species, where basically if the species can't disperse to its suitable habitat, well, we will not find it there. So you can see here the problem of using distributional data to reconstruct species ecological niches. And when we consider fossil taxa, the spatial biases are even higher because we have the problem of fossilization that is going to truncate the real distribution of the species. And we also have the problem that the sampling is very uneven and will not gather the entirety of the distribution. However, when we're interested in the species' ecological properties in environmental space, we might not actually need the whole original distribution. We rather need one that is representative of all the environments occupied by the taxon. So here, I just wanted to illustrate that point with these two graphs where we have a very kind of what we usually get in environmental space whenever we are trying to reconstruct the fitness of a species related to, for example, temperature or whatever. We usually want to get those very nice bell-shaped curves. So if we have a distribution that can account for different points of that bell-shaped curve, then we might be able to have a reconstruction that is not, I mean, that is pretty good actually. If we have occurrences that are very packed in the environmental space, so that only account for a small range of the whole potential range of the species, then it is very hard to know how we can think about how the species will be surviving in these other conditions that we're not capturing with the distributional data. So basically, our idea was to use an algorithm that is used a lot by ecologists to select sampling sites that are evenly distributed across geographic and environmental space. And this way, we can try to have the broadest view possible of all of the environments where the species could have been found. And so see if it has been found or not there. So in this study, we twist biosurvey to guide paleontological surveys toward these unsampled climates. Why? Because we have unevenly distributed research activities drawn by geological context mainly, but also research focus on specific regions. Here you have a map of the Pleistocene Africa record, where we can see that it's very concentrated in certain parts of Africa. So the idea of using biosurvey to do that was to assess coverage of continental ecological diversity. Then it will also help us restrict remote sensing and surveys to areas with a higher probability to record different paleoecosystems. And finally, it can also provide a climatic background to discuss past faunal provincialism. So now onto the study design. So we decided to focus on the Pliocene in Africa. For that, we used some climate data from the EcoClimate dataset, which models the Pliocene mean climate with 3 different baselines for downscaling. And then to see, well, how our sampling sites compared with the actual distribution of fossils, we used all mammal occurrences from 2 large paleoecological databases, so NOW and the Paleobiology database. The workflow that we used was basically, so we take those data, this environmental data and the region of interest, so here Africa. Then we process this data to make them into a master matrix of principal component analysis for the different variables. And then what biosurvey will do is that it's going to divide the environmental space into blocks. And within these blocks, it's going to select evenly distributed points within the block that will also be evenly distributed in geography, or as much as possible. So what we did is that we run this selection 100 times so that we get approximately 2,000 selected sites. And then we summed all of these selected sites to have this sort of sum density of like more frequently selected sites. So it means that all of these density spots represent climatically unique and evenly distributed climate. So applied to our context, we found those different potential survey sites that are here. So as you can see, the black points are all selected sites, but the yellow dots are the top 10 percentile selected sites, so the sites that are more frequently selected. And it's really useful because we can find like specific areas that seem to have very different climates. So this is exactly what I was saying. We find all of these spots that are unique and evenly distributed, both in environmental space and geography. And as you can see, the mammalian fossil sites do not cover all of those spots. So especially we have very few sites, for example, in these spots, even also in these spots. The Madagascar area is also very underrepresented by fossil sites. And also what's very interesting and that we started thinking about is that we can find some fossil sites in areas that were not selected by the algorithm. So how did we manage those? We actually wanted to know to which climates they corresponded, because if those areas are evenly distributed and representative climates, then it means that these fossils represent something that is close to one of these clusters in environmental space. And as you can see, we can see that those shadient sites, for example, they're very much close to the climates of this area of Africa. So now to conclude, paleobiosurvey allowed one to identify paleoenvironments sampled by the paleontological record. We applied it to the Pliocene in Africa to reveal that some underexplored areas correspond to unsampled climates. And the next steps will be to perform a sensitivity analysis based on input climate data. We would then publish the code and integrate paleobiosurvey tools to the biosurvey package. And we hope to have a publication of this work in the next special issue of the Biodiversity Informatics about biosurvey. So thank you very much for your attention. And on the name of my team, I'm happy to answer any questions that you might have.